We've seen the various surface kind of appendages that can come off the surface of our prokaryotic cells and a little bit on our eukaryotic cells. So the glycocalyx and then all these different kinds of things like the fimbriae, the extracellular matrix, and then the flagella is how we ended up looking at some of the prokaryotic cells and that idea of taxis. Well, the other major structure that we can find on the surface of many of the cells out there is what's known as the cell wall. And most of your prokaryotic cells that are out there are going to have a cell wall structure to help provide some protection for the cells that are out there. And when we look at the structure of this, most of your bacteria cell walls are going to use a molecule that's known as peptidoglycan. And when you look at the structure of peptidoglycan, think like a fence. It's going to have these boards right here that are made up of two kinds of sugars, N-acetylglucosamine and N-acetylmuramic acid, or NAG and NAM. So think long chains of this NAG and NAM right here that are attached together by polypeptide chains. So if you look at a wood fence, think of these as the boards, and then your protein chains are the cross pieces that are holding everything together right there. And if we think about this, this is all covalently bonded in a three-dimensional structure. So this is going to give the cell a whole bunch of strength on the outside and prevent them from blowing up easily. Also, when you look at this, think sugar, protein. This is food for the cells too. So again, like the glycocalyx, the peptidoglycan can be the cell storing up some excess stuff for later on. When you look at your gram-positive cells, remember they have thick peptidoglycan layers that can be up to 60 nanometers thick, and embedded inside of that peptidoglycan layer, you're going to find some teichoic acids which have phosphates associated with them. And if you remember from the chemistry chapter, phosphates have a negative charge. They're ionic. And that's where our basic dyes are actually able to attach to a gram-positive cells. Your crystal violet is actually able to attach to these long chains where you've got all these phosphates there that have the negative charge on the surface of your cells. When you look at this, here you can see an acid-fast bacteria. So not only do you have that thick peptidoglycan layer, you've also got these mycolic acids that are waxy on the surface. And if you remember from the acid-fast stain, that is trapping our carbol fuchsia molecule and giving us the hot pink color that we see right there. And so this is found with certain kinds of pathogenic bacteria. So again, things like tuberculosis are going to have this waxy layer that's going to help those cells to resist dehydration and allow them to survive for up to a month on something like a door handle or floating around in a room when somebody with TB is coughing. So again, that idea of aseptic technique and wearing masks when you're around a patient is really something that we need to be aware of. And hopefully, again, you take that message home from this class. When we look at our gram-negative cells right there, remember they have a thin peptidoglycan layer on the outside surface of the cell right here, and then surrounding that you've got an outer membrane right here that is made up of lipids and also lipopolysaccharides. So think fat and sugar right there. And what happens here is you get this sort of space in between the outer and the inner membrane right here. And this is what's known as the periplasmic space right there. And cells are going to use that to store various molecules. But remember, when you look at membranes, those are semi-permeable membranes that we'll take a look at in a second. So cells are going to have embedded in the membrane various protein channels that will allow the movement of some molecules into the periplasmic space before they can actually end up inside of the cell right there. And if we look at the exterior membrane here, or outer membrane, you can see this little molecule right here is what's known as lipid A, and it's got these sugars on the outside and then this lipid that is going to basically penetrate into that outer membrane layer right there. And this is something that is actually toxic. So your gram-negative cells can actually be more harmful to us. And what happens is if you take antibiotics and you kill off the bacterial infection, that is going to result in this outer membrane breaking down, which is going to release this lipid A molecule over here. And that lipid A molecule can result in fevers in a patient that can actually result in death. It can also result in disseminated coagulation of protein. So think your blood starts to thicken to the point where it's no longer going to be pumped productively by your heart. So even if you get rid of a gram-negative infection, the chemicals released from the cells that are breaking down can kill you off there. So distinct differences when we look at gram-negative and gram-positive cells in terms of the structure, but they still have the same basic characteristic in terms of that peptidoglycan layer on the surface of the cells is giving tensile strength to prevent the cells from growing up too much, as we'll see in a second when we look at this. When we look at our archaea cells, they also have this cell wall structure on the outside, but it could just be a protein-based thing, and so it's known as an S layer over here, or a surface layer of proteins with something, an archaea cell known as Pseuphilobus, and other bacteria that are out there are going to have something that is kind of like peptidoglycan, and they call it pseudopeptidoglycan or fake peptidoglycan. And this one has the NAG molecules, but instead of NAM, it's going to have a slightly different sugar in the chains right there. So they are made up of NAG and NAT sugars, and you still have your peptide cross bridges. So the pseudopeptidoglycan can be found with archaea cells. Again, this is a difference that distinguishes the two domains of bacteria from the domain archaea that are out there. Structural differences that are used to distinguish between these groups of microbes that are out there, in addition to something like your ribosomal RNA genes that are used to characterize them. When we look at our eukaryotic cells, they can also have a cell wall around the outside. And I found this picture here to show you, when you look in the fungus kingdom, their cell walls are made of a molecule known as chitin primarily, which is actually just long chains of N-acetylglucosamine. So they've taken that same kind of sugar molecule that your bacteria cells are using and modified it for their own uses. So they're just making long chains of NAG, and you can also find a molecule known as glucan, which is basically just long chains of sugar. So this is what we would find on the outside of fungi that are out there. And again, this is how we can place members in the fungus kingdom, because they all have this similarity in terms of their cell wall structure. And the cell wall on the outside of a fungus here is distinctly different from what we find with algae and plants that are out there. There, the cell walls are typically going to be cellulose, which is just long chains of glucose sugars. And when you look at the algae, some of them can actually use the polysaccharide agar, the stuff that we use in lab to solidify our liquid broth cultures, and others use a protein known as carrageenan, which you might find as an ingredient in your food because it actually is a thickener and causes your food to thicken. And sometimes people will add this to things like ice cream, and it actually changes whether or not your ice cream melts, and you can put your ice cream in the fridge as opposed to the freezer. The problem is your ice cream just tastes like crap then, and the consistency is off because it's that creaminess from the frozen ice cream isn't there because you're adding this artificial thickener to cause that to be sort of that semi-solid structure there. When we look at the cells that are out there, we've been looking outside the cells this whole time. We haven't actually gotten into the surface layer for the cells here when you're looking at the plasma membrane. And if you remember from the chemistry lecture, I talked a little bit about the phospholipid molecule right there that is amphipathic. The outside of the cell and the inside of the cell with the cytoplasm are both aqueous environments. So you've got water here and you've got water here. And the polar part of your phospholipid is going to make up the outside of this phospholipid bilayer there. And these phospholipids assemble spontaneously in water because the polar parts like to be in contact with water, and then the hydrophobic fatty acid tails are going to be the interior right there. And so this membrane can actually assemble by itself and is one of those ideas when we look at the spontaneous generation of some of the ancestral cells. Well, if phospholipids can form little sacs by themselves, maybe that's the origin of the protocell, or the first type of cell out there was this phospholipid bag that formed this really primitive type of membrane, and that is where we get cells evolve from these kinds of structures. And so when you look at this, remember the exterior sides are hydrophilic where the water is, and that's polar interactions with polar water. And then the interior layers of these things, you're looking at the hydrophobic fatty acid tails. And when you look at this, we can also have various things that can pass through here. And we have protein channels that are going to allow certain molecules to get through this membrane, because remember, membranes are semi-permeable. They allow small uncharged molecules to flow through here, but if you have a charge, charges are going to hit this non-polar fatty acid region right here, and they're just going to bounce right off. So something like a sodium or a chloride ion cannot pass easily through this membrane, and so that's where we've got these protein channels that can open up and allow these molecules to travel back and forth across this. And sometimes you'll find proteins embedded in membrane that are on the periphery, so they're known as peripheral proteins. And you can see there's one on the exterior and there's one on the interior of the membrane. And then you've got these transmembrane proteins that go all the way across the membrane right there. And those are the ones that could potentially allow signals to be transmitted from the outside to the inside, or allow molecules to move across the membrane, because you can open up a gate or a window in the membrane right there. When we look at this process of this, notice there are other, or when we look at the anatomy of this plasma membrane, you can also see these other kinds of sugar proteins and sugar lipids or glycolipids on the surface here. There's your extracellular matrix. There is things like a glycocalyx that are tethered in the membrane right there. And when we look at the phospholipid structure that is out there, one big difference that we see with archaea is a very different form of phospholipid structures. And we'll look at that a little bit more in chapter 4 when we get to that stuff right there. With eukaryotic cells, you do have this phospholipid bilayer, and you've got these embedded proteins, and when they measure it, it's actually about a 50-50 mixture of phospholipids and proteins, as well as these various glycoproteins and glycolipids that are on the outside there. The other thing that we can find with eukaryotic cells is buried in this phospholipid layer are steroid molecules. And steroids are pretty much hydrophobic, so they're going to dissolve and get trapped in there. And so we'll find that in things like plants and fungi have steroids. In animals, we actually use cholesterol molecules that are going to embed in there. And if you think about it, that's going to make the membrane even more hydrophobic. So if this structure starts to fall apart, water is going to impinge on there, and the presence of the steroid in there is going to allow it to basically reform, because it's going to be more hydrophobic in the membrane there. And if we look at this, we can find a variety of functions for the cell membrane. And if you look at this list here, you can see just a partial list of some of the different things I want you to think about, because we always think of the cell membrane as it surrounds the cytoplasm, and this is what separates the interior cytoplasmic fluid from the exterior or outside. But remember, that membrane is selectively permeable. It's going to control the movement of substances in and out of the cells there. If you are large or a charged molecule, you cannot move past that phospholipid bilayer there. And so that's where we saw this example of these porins and these channel proteins. When we look at the cells that are out there, you are also going to use your cell membrane to signal other cells. So think those glycoproteins and glycolipids on the surface there. Those can be used to signal to cells. And also, when you look at cells, you can squirt chemicals to the outside or surface of the cell to signal cells in the surrounding area. So you're going to have pumps that are built into your membrane to squirt these things out. When we look at bacteria cells, we can also see a variety of differences in terms of the membrane. This is actually where their chromosomes attach to the membrane, unlike a eukaryotic cell where we find the chromosomes in the nucleus. When you look at bacteria cells, they've got various proteins and other structures embedded into their cytoplasmic membrane that are going to be used for the production of energy. And also that sort of basic idea that the structures for the cells are going to actually attach to the cell membrane. In addition to something like the chromosome, you can also have cell walls or the cytoskeleton on the inside. So when you look at your cell membrane, remember it's not just a barrier. It does serve other functions for the cells that are out there.